The QuantaBio sparQ Universal Library Quant Kit is a fast, reliable, and precise solution for qPCR-based quantification of NGS libraries. You can complete the full protocol in under 45 minutes from setup to results. Achieve accurate quantification across 6 orders of magnitude. It provides a convenient, ready-to-use master mix with P5, P7 primers, and enjoys seamless compatibility with all real-time PCR platforms. The QuantaBio sparQ Universal Library Quant Kit is intended for molecular biology applications. This product is not intended for the diagnosis, prevention, or treatment of a disease. The sparQ Universal Library Quant Kit includes all the reagents you need for qPCR-based quantification of Illumina-compatible NGS libraries. The master mix is pre-mixed with primers targeting the P5 and P7 adapter sequences, so there's no need to add primers separately. The included DNA standards are pre-diluted and allow you to generate a standard curve ranging from 0.002 picomolar to 20 picomolar, ensuring a broad dynamic range of coverage. The required 10x library dilution buffer has a blue cap. For use with HYROX instruments, spin down the HYROX reference dye and add the appropriate amount of reference dye based on your kit size upon first opening of the product. As example, for the 100 reaction kit, add 5 microliters of the reference dye directly to the sparQ Universal Fast Master Mix tube. For the 500 reaction kit, add 25 microliters of the reference dye directly to the sparQ Universal Fast Master Mix bottle. Gently vortex for 30 seconds to mix thoroughly before proceeding with the protocol. Nuclease-free water is required for this step and is not included in the kit. To prepare the 1x library dilution buffer, combine 0.9 ml of nuclease-free water with 0.1 ml of 10x library dilution buffer in a clean tube. Mix thoroughly by vortexing. This volume can be scaled up or down depending on how many samples are being run. Plan for about 0.4 to 0.6 ml of 1x library dilution buffer per sample. Optimal dilutions for qPCR may vary depending on the nature of your NGS sequencing operations. It is important that one of the library dilutions fall within the linear dynamic range of the supplied sparQ DNA standards. A general guideline to prepare the serial dilution is to first prepare a 1 to 100 dilution by adding 198 microliters of 1x library dilution buffer and 2 microliters of the library sample in a 1.5 ml tube. Mix by vortexing and briefly centrifuge. Next, prepare a 1 in 10,000 dilution by taking 198 microliters of 1x buffer and adding 2 microliters of the 1 in 100 dilution, vortex, and centrifuge. Finally, for the 1 in 100,000 dilution, add 45 microliters of 1x buffer to 5 microliters of the 1 in 10,000 dilution, vortex, and centrifuge. Run all standards and library dilutions in triplicate for optimal accuracy. Prepare a 96-well plate by labeling it and organizing the layout, ensuring it's clean and ready for qPCR setup. An example is shown here. Keep all reagents on ice during setup. The required sparQ Universal Fast Master Mix has an orange cap. Dispense 16 microliters of sparQ Universal Fast Master Mix into each well. Add 4 microliters of diluted library sample or sparQ DNA standard to the corresponding wells. For smaller reaction volumes, scale components proportionally. Seal the plate then briefly vortex to mix and centrifuge to collect the contents at the bottom of each well. Pre-program the thermocycler according to the table and preheat the block to 95 degrees Celsius. Begin by confirming that the reaction plate is properly annotated in the instrument software. Add your samples, then start the run. Download the companion NGS calculator from the QuantaBio website. This tool allows for easy calculation of the concentration of unknown libraries. Begin by entering the sparQ values for the standards. The calculator generates a standard curve plot and calculates the reaction efficiency. Next, enter the dilution factors for the NGS or unknown libraries found in cell D79 and D80. Input the average fragment length values for the unknown libraries into column E. Then, add the sparQ values for the libraries into column F. The library concentrations are automatically calculated, and the results can be found in columns M, N, and O.